Hello everyone. Welcome to today's live broadcast, SPR for aptamer-based molecular interactions in programmable materials. I'm Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Reichert Technologies Life Sciences. Reichert develops and manufactures precision analytical and diagnostic equipment in Buffalo, New York, including surface plasmon resonance, SPR, instruments, which have become known for their robustness, flexibility, ease of use, and outstanding sensitivity. Please visit www.reichertspr.com for more information on Reichert SPR instruments. Additionally, Reichert's solution team is happy to address any questions and discuss how we can help you overcome your interaction analysis challenges. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of your presentation window. We'll try to answer as many of your questions as we can today. You can enlarge the slide window by clicking on the screen icon in the lower right of the slide window. If you have any technical problems hearing or viewing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Erin Gaddis, PhD candidate, Penn State University Department of Bioengineering. I will now turn it over to Erin for her presentation.
Hello, thank you for the great introduction. I'm Erin Gaddis. I am a PhD candidate at Penn State University working in the Wong Lab. So our lab's focus is on using oligonucleotides and aptamers to create biomolecular and biomimetic materials. So we use surface plasmon resonance in evaluating a lot of the binding affinities and the interactions that are so pivotal for these materials. So this talk is going to focus on how we use SPR, but also we'll have a more broad reach into how SPR functions and some other samples and techniques that can be used for SPR. So first I'm going to introduce what surface plasmon resonance, or SPR, is and how it works. I'm also going to be looking at the samples and detection strategies that are available, and then talk a little bit about data analysis and how this can be done in the output from SPR. Next, I'm going to be talking a bit about how our lab uses surface plasmon resonance to evaluate biomolecular interactions, specifically using oligonucleotides. So we do this using oligonucleotides for cell capture and release, for the loading and release of growth factors, and also for signal amplification. So surface plasmon resonance is a technique that is able to evaluate the molecular interactions between two molecules in real time and also in a label-free environment. And the surface plasmon resonance system really functions with three main components, the first being the sensor chip, which contains the immobilized ligand of interest. Next is the microfluidic system, which delivers the analyte to the sensor chip. And then the third and probably most complex part is the optical measurement system, which allows the changes in local refractive indices that are sensed to indicate the changes in mass at the surface chip interface. So you can see in the image on the upper right our lab's surface plasmon resonance system that has a dual channel, so a sample and reference channel. And the image on the lower right shows kind of the sensorgram schematic for what we're looking at for SPR, where we have a ligand immobilized to the sensor chip. We have an analyte that's able to bind to this ligand. Eventually, most of the reaction sites on the ligand chip are filled, and then we have some dissociation of the analyte from the ligand. So first I'm going to talk a bit about the optical detection system. This is probably the most important part for understanding how surface plasmon resonance works. So the optical detection system functions based on the use of a prism and the sensor chip above this prism. The sensor chip is coated with a thin gold film, and the prism has a refractive index that is higher than that of the environment at the surface interface of the chip. So this is something like a physiological buffer. So under these conditions, when light enters the prism above the critical angle, it can be totally internally reflected. And this reflection and the photons that are involved in this produce an evanescent wave at the interface that decays as it goes away from the interface. About 300 nanometers is as far as it extends. So when mobile electrons in this thin gold layer at the interface are moving, we can think of that more like a plasma surface since they're mobile. And surface plasmons from the density fluctuations at this interface can propagate along the surface. So when we have light that has the momentum of photons matching that of the surface plasmons, we can say that resonance occurs. And this is based on some parameters such as the angle of light, the wavelength of the light, and also the refractive indices of the materials, the prism and the solution. So when the photons from the light are exciting the plasmons, we can note that there's a reduction in detected light since these photons are no longer being reflected and detected. And this reduction occurs at a specific angle. The minimum of this reduction is at a specific angle, which we can call the resonance angle. So when we're scanning a range of angles of light, we can look at the reflectivity and see that a minimum exists, and this is our resonance angle, as you can see in the figure on the left. So when the output reaches this, we have the minimum, and we can see that this shifts as we have mass that's absorbed on our sensor chip. So changes in the interface are going to change the local refractive index, and this is what's altering the resonance angle. So now that we've looked at the optical detection system, we can focus on the sensor chip. So the sensor chip, as I've explained earlier, consists of a thin middle layer that is on top of a glass chip. And this allows for the ligand to be immobilized, and this is the ligand that you're looking at interactions with. So some immobilization strategies are listed in this slide, the most common being amine coupling using EDC-NHS chemistry. And this forms a covalent bond. Some other strategies are using a streptavidin or a neutravidin on the chip and a biotinylated ligand. And also gold thiol can be used to create self-assembled monolayers. So there are a variety of immobilization strategies. And this is important to know because when we're doing immobilization, it may be more advantageous, depending on your ligand, to use a different one of these strategies. And also, we can think about the amount of binding sites we have on our chip. So many chips use a polymer matrix to increase the amount of binding site at the chip. So we can look at the balance between binding sites and signal strength in the image in the center of this slide. We can see that as the evanescent wave moves from the interface, we can see it decays. So at 300 nanometers, there's a very low signal strength, whereas at the interface there's a much higher signal strength. So binding interactions that occur further from the surface actually will have less of a signal that's being detected than those that are near the interface. So this binding balance between the binding sites and signal strength is usually around 100 nanometers, which is the polymer length from the surface generally. We can also look at the figure on the left, which shows a sensorgram for an amine coupling reaction for the immobilization of a ligand, or several ligands in this case. We can see in A, first we have a baseline sensor signal, which at B increases as we use EDC-NHS to activate the surface. At C, we see that the baseline is maintained after we are washing the surface with our running buffer. And then in D, we incubate this surface with the ligand of interest, or in this case, several ligands. We can see the mass increasing, and then in E, we do a wash step to remove the the binding of loose ligands. In F, the group in the scientific reports paper here uses a deactivation step which deactivates the surface but also is used to remove loosely bound ligands. And then in G you can see that the final signal is higher than that in A, which would show that the ligands have been immobilized on the surface. So when we're looking at a flow cell like that on the bottom right, we can see that it sits atop of our sensor chip and has two channels, the first being the immobilization channel or sample channel, where the ligand is immobilized. In the second channel, we have a reference channel where there's no ligand, and this allows the microfluidic system to be able to deliver analyte to both channels in equal conditions, with the exception of one having a ligand and one having no ligand. And this serves as a control that can be subtracted from the sample channel later for data analysis. So after we have bound our analytes of interest and allowed their dissociation, we generate binding curves, and now we need to analyze them. So in Reichert software, the analysis is done using TraceDrawer, which is shown in the screenshot on the top image. And this allows for the alignment of start time and response to 0, blank subtractions and subtractions of reference channel, and then the fitting of the curves that are generated to models, such as 1-to-1 binding, 2-to-1 or 1-to-2 binding, and then allowing for some considerations like mass transport depletion, which is shown in the lower image. And this occurs when the bulk concentration has a higher concentration of analyte than that in the what we call a depletion zone or layer, right at the interface. And this has a slower association rate than would be expected based on the concentration in the bulk. And in a similar way, during dissociation, there can become a retention layer or zone near the interface where the concentration of analyte is higher than that of the bulk, so that molecules will dissociate but then rebind to another site. And this slows the dissociation rate. So these considerations can be made when analyzing data. So other than binding kinetics, like that shown in the image on the top, we can do other types of analysis using surface plasmon resonance. This includes equilibrium analysis, like the lower image, where the concentration is plotted against the maximum response. We can also look at the specificity of molecules binding with immobilized molecules. And we can look at many different types of molecular interactions, such as the interactions between proteins and small molecules, cells, artificial membranes, and oligonucleotides. So our lab specifically looks at oligonucleotide interactions, either with other oligonucleotides or in some cases proteins. So our lab also focuses on the use of oligonucleotide aptamers. And I'll give a little bit of background on what aptamers are. So nucleic acid aptamers, they're short single-stranded oligonucleotides that can be selected from randomized libraries using the SELECT process. So the SELECT process is shown on the right. In this process, the randomized library is incubated with the target of interest. And this can be a cell or it can be a protein. And the nice thing about this process is if you have a cell of interest, you don't have to know the specific surface receptors you're looking at. You can select toward this cell and then later determine what the target on the cell actually was. After selecting against the target of interest, this population that binds is later allowed to interact with a control target or a negative target. And those, the sequences that do not bind to the negative target are then selected, amplified, and brought through another round. So as the SELECT process occurs round after round, the population of interest and with high binding specificity to the target of interest is enriched. And this is how aptamers are developed. So some of the benefits of using aptamers, especially when we're comparing aptamers with other molecules like antibodies, is that aptamers are very small. They're usually less than 50 nucleotides in length. So they're very simple to synthesize using chemical methods with little batch-to-batch variation, which is helpful when conducting experiments for this process. And then when we're looking at aptamers, we see very little immunogenicity. So this is good for biomolecular interactions. We don't have these types of concerns for biocompatibility. Aptamers are also small so that they can be more easily delivered intracellularly. Our group uses surface plasmon resonance in many ways, but also other groups are very interested in using surface plasmon resonance to look at oligonucleotide interactions with other biomolecules. So some of these applications are listed here. Biosensors is a great application for surface plasmon resonance and oligonucleotides. So in the central figure by Chen et al., we can see that a VEGF aptamer is immobilized on the surface of the surface plasmon resonance sensor chip, and then VEGF is able to be bound to this. In the next step, another aptamer for VEGF that binds a different site is allowed to incubate, and this aptamer contains multiple primers that can be extended to increase the mass on the surface and detect very small quantities of VEGF. We can also see that surface plasmon resonance can be used to evaluate the affinity of aptamers for their targets. So in the figure on the right, we can see two aptamers that are specific for thrombin, which is shown in the center. These antithrombin aptamers bind to different locations on thrombin, one being the fibrinogen binding site and the second being the heparin binding site. In this study by Hasegawa et al., we can see that they use a linker to link these two different antibodies, and they evaluate the binding affinity of this aptamer dimer for thrombin. Aptamer affinity has been studied for many different aptamers, some of which I've listed below. Next, we can see that aptamers can be used to verify the specificity of the sequence, and also to look at some of the ways that sequences interact with each other, such as the formation of DNA polymers, or the displacement of oligonucleotides by other oligonucleotides. So at this point, I'm going to move on to some of the work that our group has done. We look at the specificity of some sequences using SPR in this figure. So you can see in the cartoon on the left, we have a sequence A that's immobilized on our on our chip, and we usually use neutravida and biotin interactions in this case. So the biotinylated sequence is immobilized, and then we incubate this chip with sequence B, which binds to sequence A with 20 base pairs. And when we look at the sensorgram on the right, we can see that sequence B is able to bind in situations when the sequence has not been altered, but when the sequence has been scrambled, or the order of the nucleotides has been randomized, we can see the blue signal showing that very little binding is occurring. So the sequence specificity is very important in this case. Next, we can use sequence C, which binds to sequence B with 25 base pairs, to dissociate sequence B from A. And this you can see in the sensorgram in the red signal, where the sequence has 25 base pairs. If we lower the amount of base pairing in C to only 20 base pairs, we can see that this displacement does not occur. In a similar way, if we scramble the sequence of C, it doesn't specifically bind to B in this case, and in the same way, we don't see any dissociation of B from A. So we're going to look at an application for this system using these sequences to release captured tumor cells from a surface. So in the schematic on the left, we can see that a sequence, the primary complementary sequence in purple, is attached to a hydrogel surface. And an aptamer is able to hybridize with this sequence, much like sequence A and sequence B were able to hybridize in the previous slide. These aptamers are specific to a receptor on a tumor cell line, which is shown being captured on this surface in the images on the right. We can capture these cells and then quantify the amount of cells bound per area. Next, we can use a secondary sequence which binds to the aptamer sequence with more base pairs than the aptamer sequence binds to the primary sequence. And this displaces not only the aptamer sequence, but also any cells that are attached to this aptamer. And when we look at the images back on the right, we can see that when the complementary sequence is 25 base pairs with the aptamer, most cells are released. When the sequence is scrambled on the rightmost image, we can see that very little cells are released. So this is a very specific release system. And this sequence, or in this system, we can see that the surface can be regenerated for multiple rounds of cell capture and release. And this is important when we look at how tumor cells are able to be captured and then evaluated. So tumor cells are often very rare and difficult to isolate. So once they're captured, it's important to be able to remove them in a non-destructive way and then analyze the surface markers that may be used to create more effective drugs or targeting strategies in the future. We can also use surface plasmon resonance to evaluate the binding affinity of aptamers versus their length. So in the image on the left, we can see the sensorgram showing that different lengths of aptamers have different binding affinities, and that we can use, yet again, a triggering sequence on the right to release these aptamers from their target. So we can apply this to a PDGF-BB release system. So this is an aptamer that is able to bind to this protein. We can create 3 different versions of this aptamer: the high affinity aptamer, the moderate affinity aptamer, which is the same as the high affinity except for 2 nucleotides that have been altered, and then the low affinity aptamer, which is much more different from the high affinity aptamer. And we can see that each aptamer has a specific secondary structure based on the sequence, and that the changing the 2 nucleotides from the high to the moderate affinity aptamers vastly alters the secondary structure. And this is going to affect how the aptamer is able to bind to its target. So we can use SPR to evaluate the binding of this aptamer with its target at different concentrations. And when we look in the graph on the right, this sensorgram shows that the binding affinity of the high is much more than the moderate and low aptamers at the same concentration. So when we apply this to a system, we can use this aptamer to load and then also release PDGF-BB from a biomaterial, which in this case is a hydrogel. So this hydrogel is porous and can be loaded with the protein of interest. In this case, the aptamer is already conjugated within the hydrogel matrix, so it's chemically conjugated, not physically conjugated. And then we can load this protein of interest, and it forms a complex with the aptamer, so it's binding specifically. And we can see in the graph on the lower left that the highest affinity aptamer is able to load the most amount of PDGF-BB. Next, we can use a complementary sequence, which hybridizes with our conjugated aptamer sequence to free the target protein. And this is a sequence-specific and dosage-specific release. So we can see that when 1x amount of our complementary sequence is added, the release is only half of that of a 2x dosage. And this can be done at multiple periods of time to release the PDGF-BB on demand. We can also use SPR to evaluate the formation of DNA polymers from the surface of a chip. So in this case, we'll have a linear DNA initiator structure, or DI, which is immobilized on the sensor chip, which is not shown in the sensorgram on the right. Next, we incubate 2 DNA monomers, DM1 and DM2. DM1 is able to bind to the initiator and change the conformation from a hairpin structure, as shown on the left, to a linear structure. In this case, the linear structure serves as an initiator for the hairpin structure of DM2 to open to a linear structure, which initiates DM1. So this will alternate sequentially to form a DNA polymer. In the sensorgram on the right, we can see that when the initiator is immobilized on the chip and we pass DM2 over the chip, this monomer cannot interact with the initiator to linearize, and therefore very little binding is observed. When DM1, however, is incubated with this initiator functionalized chip, we can see an increase in mass, which would indicate that the monomer 1 has bound to the surface. When both monomers are incubated in solution, we can see a much greater increase in mass, which would suggest that the polymerization is occurring at the surface. So this system can be used as a signal amplification strategy. In this instance, we use nanoparticles that have an attached DNA initiator as an activation point for the polymerization to occur. So in the left image, the schematic one shows that when there's no initiator, the polymerization cannot occur on the particle. In the sequence number 2, or in number 2, we can see that the initiator can be bound by monomer 1, but we can't see a large increase in fluorescence in 2 for the fluorescence image at the lower area. However, in 3, we can see that when both monomers, which have a fluorophore conjugated to them, are able to be incubated with the initiator functionalized particle, a large amount of fluorescence intensity can be observed. So this shows that the use of the polymers with the fluorophore attached to each monomer can create a larger fluorescent signal than one fluorophore per reaction site. And this can be observed when we're looking at labeling biomolecules. So in this case, collagen is being labeled. And on the right, we can see that when each monomer is labeled by a different fluorophore, we can see that they can be co-localized, which would show that not only are the monomers binding to the particle, but also they're in the same location and would indicate the formation of DNA polymers at these locations. We can also functionalize our DNA polymers with aptamers to create polyvalent aptamers and increase the amount of affinity ligands that can be conjugated per reaction site. So in this case, we have an initiator, which, similar to the last study, we can be using as a starting point to form DNA polymers, which can be our polymeric scaffold. In this case, though, we have a binding region that's on a monomer 1, which we can call the toehold region. And this extends and allows the binding and hybridization of the aptamer. So when the aptamer is incubated with this polymeric scaffold, binding between the aptamer and this toehold region creates a polyvalent aptamer structure. So when we look at the sensorgram on the right, we can see that the DNA polymers can be formed through the use of both monomers, and that by having this binding region, in the case of the yellow versus the green line, we can see that the aptamer can be bound when there's this toehold region, whereas the aptamer cannot be bound in the case of a monomer 1 with no toehold region. When we have a polymer scaffold, we can see in the inset that the aptamer injection results in a larger increase of mass, which would show that polymeric and polyvalent aptamers have been formed. And this occurs relatively quickly, less than 2 minutes. So we can use this polyvalent aptamer system to increase the ligand display for the capture of circulating tumor cells. So in this case, the aptamer is specific for a surface receptor on a circulating tumor cell line. And we can compare the surface reaction sites with one aptamer per reaction site, which we call direct conjugation, to multiple aptamers per reaction site in our polymeric display. And this is done on a hydrogel surface. So we can see that as the cells are incubated for longer amounts of time, the amount of captured cells increases in both cases. However, the polyvalent display affords a larger amount of captured cells at each time point. And this is a significant amount of cell capture. So this is important for areas that have very difficult surfaces to functionalize or where there's limited surface reaction sites available. We can also release our DNA polymers from the surface using a complementary sequence, similar to some of the complementary sequence releases we've seen earlier. So in this case, we create DNA polymers on a surface and then incubate these polymeric scaffolds with aptamers to create polyvalent aptamers. And we can see in the sensorgram on the left that when the complementary sequence that's specific for this polymer has been incubated with the polyvalent aptamers, a large decrease in mass is found to occur. However, when this complementary sequence is scrambled, we can see that the mass does not significantly decrease. So we can see that the complementary sequence is specific for this system. When we look at the figure on the right, we can see that when our aptamers are labeled with a fluorophore, our DNA polymers can be shown as a fluorescent image, and that cells can be captured. When we add a scrambled complementary sequence, we see very little decrease in fluorescence intensity, and most cells remain bound to our hydrogel. However, when we use our complementary sequence in its functional form, we can see that the fluorescence intensity of the hydrogel decreases significantly, and this would indicate that the DNA polymers have been dissociated from the surface. When we look at the amount of cells bound in this instance, we can see that many have been dissociated as well. And this is important to note that this is not from the complementary sequence interacting directly with the cells, but instead by dissociating the DNA polymers. So as a summary, today we've talked a bit about how SPR can be used to detect molecular interactions in real time and with no necessary labeling steps. We've also looked at the types of samples that can be used, including proteins, nucleic acids, and other small molecules, and how aptamers can be used to test the binding of targets with tunable affinity. We've also talked a bit about how our lab group uses SPR to evaluate the aptamer specificity, the ability to release aptamers, and how DNA hybridization can be used to form polyvalent aptamers and DNA polymers. If you're more interested in learning a bit about surface plasmon resonance and the fundamentals about this process, these are some resources I recommend, the first two bullets. Additionally, if you have any, if you see the use of SPR in your future, I would recommend looking at Reichert technology, which is the system that we use. Also for the analysis software, I would recommend looking at TraceDraw, which is associated with Reichert software, and also with Scrubber, which is another data analysis software. I'd like to thank my advisor, Dr. Young Wong, and all of my lab mates in the Wong lab, especially Mark, Nyesao, and Shiwi, whose research has been featured heavily in this presentation. I'd also like to thank the host, Reichert Technologies, for giving me this great opportunity, and all of the funding sources that allow this research to be conducted. So at this point, I'd be happy to take any questions you may have. Thank you.
Thank you, Erin, for that informative presentation. Before we get started on our question and answer session, I'd like to remind our audience how to submit questions. You can submit your questions by typing them in the Q&A button, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many of your questions as we can. We'd also like to let our audience know how to claim their continuing education certificate. Please click on the CE button at the bottom left, which will take you to the AACC Accent page to log in and claim your certificate. Our first question is, how do you deconvolute the specific interaction of an aptamer to its target from the non-specific ones?
This is a nice question. It's important for aptamers to be specific for their target, and that's why the SELECT process has a very rigorous amount of cycles and negative target selection steps. And this allows the target to be selected only for those molecules that you're wanting to bind. And this can be tested later using SPR for control molecules as well. But a lot of the specificity is determined using the SELECT process. And from there, a range of aptamers can be determined and tested for further binding specificity.
Great, thank you. Our next question, how do you screen off the negative charges from an aptamer's phosphate backbone in causing non-specific charged interactions?
So The buffer that is used when working with aptamers is very important to diffuse some of the repulsive negative charges, and this is what's allowing the DNA, specifically for something like a hybridization, to occur. Otherwise, DNA sequences may not be able to hybridize and form these secondary structures or polymers. So buffer is very important in this regard.
Thank you. Our next question: have you investigated the different effects of cations, particularly divalent ones, to aptamer binding?
So divalent cations are they're important and are especially associated with a lot of aggregation and binding. And this work has been done by a lot of groups, especially when looking at buffers and the importance of these cations in the binding of aptamers to other biomolecules. But our group does not specialize so much in the select process in regards to this, and more so in the testing of aptamers to create biomaterials, functional biomaterials.
Thank you. Our next question: in your experience, do you get a lot of background binding with streptavidin biotin systems?
So we often work with biotin-streptavidin interactions for SPR, and we find that usually we don't have a lot of non-specific binding in this instance. The biotin-streptavidin or neutravidin bond is very strong, so that's an advantage when we're immobilizing our ligands. And we don't find that we have a lot of extra binding to the surface in this instance.
Thank you. We have so many good questions. Our next question: how do you distinguish the binding of an aptamer, whether it's based on structure or sequence?
So the aptamer specificity comes from its sequence, and that's what allows it to form these secondary structures that allow it to interact specifically with its target. So finding these sequences is very important. And after using the select process, we can sequence the sequences that have been found to bind the most specifically toward our aptamer, or toward our targets. And in this case, we can discover a bit more about the binding interactions between our aptamers and our specific targets, and maybe even the identity of these targets.
Thank you. Our next question: is sequence A, B, C an aptamer?
So in our interactions shown by sequences A, B, and C, we have sequence A, which is not an aptamer, but just a binding site for sequence B, which is an aptamer. And sequence B is what is interacting with our target biomolecules, in this case, oftentimes a cell or a protein. And then sequence C is not an aptamer, but binds to the aptamer with more base pairs than between B and A. And this allows the dissociation of our aptamer from A, and allows A to be remaining on the surface so that it can be used for another round of capture and release.
Great. Our next question: do you normally immobilize your aptamer target to the SPR chip? If so, do you use amine coupling as the conjugation technique?
So when we're evaluating aptamers binding to proteins, we often use amine coupling. And we find that this is a pretty useful way of immobilizing the proteins to the surface. And usually this system has worked out very well for our analysis.
Great. Our next question: how do you control the release of growth factors from the hydrogel per time? The dose?
So the release of growth factors can be controlled in kind of two different ways. The first being the amount or concentration of a complementary release sequence, which can determine the amount of protein that is released from the aptamer that's loading this protein. In the other instance, we can use different aptamer affinities, and this can regulate the amount of growth factor that's bound, and potentially the amount and strongness of binding between these two, so that some may be dissociated. But this is a less controlled way of doing this.
Great. We have one more question. Could you mention the software for analysis again?
So the analysis software that we use TraceDrawer, but also we use Scrubber. And the data from the Riker system that we get from the binding of our analyte to our ligand can be exported into either. Both work very well.
Great. Thank you. That is all the time that we have for questions now. I'd just like to turn it back to Erin to see if she has any final comments or advice for our audience.
I guess I would just like to say that SPR has been a great technique that our lab has used in many different types of studies, and we've gained a lot of quantitative information about the interactions between our oligonucleotide systems and their target molecules. But if this is the type of interaction you'd like to investigate, even if it's not with aptamers, I would highly recommend looking into using surface plasmon resonance to get a bit more of the binding interaction data in your systems. I think it's a very worthwhile technique to attempt for any biomolecular interactions.
Thank you again, Erin. I would also like to thank today's sponsor, Riker Technologies Life Sciences, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through March 2016. You'll be receiving an email from LabRoots alerting you as to when that webcast will be available for replay. And we invite you to forward that email to your colleagues who may have missed today's live event. Once again, thank you to Erin and to our sponsor, Riker Life Technologies. Thank you very much.